All right. Okay, so we are here to talk about, I guess. All right, so we're returning to the synapse. And so we talked in the prior sections basically about the membrane and action potential. And all of this is kind of leading to some of this synapse action, because this is where like the knowledge transfer occurs basically. So, so let's go through this basic structure. So the presynaptic membrane, this is basically right here. It's at the axon terminal, which is basically the end. And you can kind of see this presynaptic membrane. It kind of wraps all the way around. Okay. And then you have this basically being this presynaptic terminal, which is where some of these were released from. This space in between right here, this sort of darker area, is the synaptic cleft right in between these. And this is a small gap where actually the chemicals transfer from this guy to this guy. And the postsynaptic membrane right up here, this is also like the receiving side of the chemical messengers. So this is basically like EPSPs or IPSPs are being generated. So this action potential, so this sort of at the end of this, then this is sort of excitatory postsynaptic potentials or inhibitory postsynaptic potentials that would then cause this cell to basically be excitatory or inhibitory. So they would pass, that would be here, and it would pass through the synaptic cleft. And then you have these synaptic vesicles right in here. Some of these, right here, basically. These little blue ones here, they are small membrane bound spheres that contain the neurotransmitters. So this is actually how the neurotransmitters are brought down. These small synaptic vesicles actually travel down the axon on these little filaments, and they sort of gather here to basically be preparing for release. You can see them all kind of clouding up down here. And so here, because this is where these vesicles would basically merge with this presynaptic membrane, excuse me, with this presynaptic membrane, and they would sort of like open up and merge with it, and that's what would release the neurotransmitters. And then you have these storage granules. You don't really see them on here, but they're small little that will actually just make, store the kind of compartments that hold several vesicles containing the neurotransmitters. So it probably some of like if these types of things, if they were wrapped up and had these inside, but they just don't have the best example right here. So all in all, at the synapse, the chemical sort of the voltage is coming down right here to the presynaptic membrane, and which was going to touch base with the postsynaptic membrane through the release of these neurotransmitters. Now, the presynaptic membrane and these neurotransmitters, once they are released, will have either an excitatory effect on this postsynaptic membrane or an inhibitory effect on this, which will then be trying to nudge the cell towards EPSP's depolarization, and IPSPs would lead to repolarization, or just hyperpolarization, making the cell more negative. So these are these parts of the synapse. And then what this would be over here, what this postsynaptic membrane would be, this postsynaptic membrane would be the connection of this axon terminal with a dendritic spine. So this is very small, what this dendritic spine would look like and connect with these up here. All right, so there's different types of synapses. And a synapse is basically any place where one neuron communicates with another. And these synapses there can be basically multiple types. You have an axodendritic, which is the terminal end of an axon communicating with the dendrites of another neuron, which is what we saw in the sort of prior little example above. Or you can have axosomatic, where the terminal end of the axon communicating with the cell body or soma of another neuron. So basically you have your cell body, and then you're going to have your dendritic, branches and spines that reach off. So axodendritic, the neuron connects with those dendritic spines. And then versus axosomatic, then it comes and tries to actually connect with the cell body. And then you also have axoaxonic, that is the terminal end basically communicating with the axon of another neuron. And so that's where those are not quite as common, but you can imagine that if the end of a neuron, like the end of the axon terminal, is basically kind of communicating with the axon, then there's going to be sort of, different types of effects of how those neurons communicate. So each of these axodendritic, which is connecting with the dendrites, axosomatic, connecting with the cell body or soma, think of these and how you can remember them, axoaxonic, the terminal with another axon. And then with these connection points, you also have, again, these dendritic spines, which would be, in the axodendritic. And these are these small protuberances along the dendrites, and they often meet the terminal buttons. And so here was a couple of, this is an axon. So this would basically be an axon, the sort of dendritic going to the axon. So this would actually be an axon axonic. And then this is basically the dendrite, and then you basically have the little spine that branches up. And this one, these are different lengths. So this one would be like a mushroom one, which is sort of stubby, doesn't reach out so much. And then you have a little stubby one right here. And technically this is also where how they would start to grow. All right. So this is not on the exam, but I'm going to cover it briefly just so you understand that there are different types of synapses. So we also what's known as an electrical synapse. And an electrical synapse is when current flows directly between the neurons, and it passes ions and molecules. So there are specific junctions here that basically allow neuron 1 to sort of have a direct effect on neuron 2 in terms of this voltage change. So this place where the neurons touch each other is called this gap junction right here. And gap junctions, are made of connexons that are embedded in the cell membrane. And these specific forms of connexons, then they're able to basically have the flow from neuron 1 sort of pass through elements of these directly to neuron 2. And these are these connexons. So when they're closed, then you're not going to have any of that direct flow. But when they open, then you're going to have this direct flow. So that's how you can also control when, the electric, when, they're not just touching all the time, having stuff, flowing freely through in terms of electricity, like neuron 1 continually stimulating neuron 2. So no, that would not be of great function to sort of the nervous system. But you have these particular sort of molecules that flows through, and where open it goes through, and when it's closed it does not. All right, so shift this over here a little bit in terms of, you guys know that this is chemical synapses. All right, so we've got a chemical synapse. I talked about this a little earlier, where I just mentioned, these, and we were going over these synapses. And the chemical synapse, any synapses generally vary widely, and each type is specialized in location, structure, function, and target. So we talked a little bit more about the chemical synapse, and so here, this is the more common than electrical synapses. And we're going to show some of the different types of connections here. So this is going to be kind of a continuation of the previous slide with more examples. And in the chemical synapse, unlike the electrical, the neurons do not touch. But then again, there is this small gap, the synaptic cleft, where this flow of information occurs. And when those vesicles open and sort of merge with the presynaptic membrane to release the neurotransmitters, that's a process called exocytosis. And when these chemical messenger neurotransmitters that they these chemical messages then sort of cause this the neurotransmitter to be released from the postsynaptic neuron, and this exocytosis is facilitated, by calcium entering the presynaptic neuron, and that kind of signals again the release of the neurotransmitters. So here's more of the different types of connections between neurons. So we went through a couple of these. So when we get to them, I'll kind of breeze through those a little bit more. But one that we haven't talked about, dendrodendritic. This is dendrites sending messages to other dendrites that they can kind of meet, right here on their spines, like two spines getting together. Then you have the axodendritic. This is what we talked about before, where you have an axon that's going to meet with the dendrite. We have axo extracellular. Okay, so this is quite different. This is a terminal with no specific target that secretes transmitter into extracellular fluid. So this would be something that might happen with how neurotransmitters that act as hormones would be released. You may have heard my dog in the background. He's woken up for a nap, and I will be taking him out for a walk after we finish this next set of slides. Then we have the axoaxomic, which we talked about. And this is where, the axon connects with the soma, the cell body right here, as you can see. And then we've got this axosynaptic, and that is where the axon terminal ends on another terminal, just as you can see here, where these then, that other neuron, sorry, my dog, has the other neuron, like in this case, could be these two connecting. If they're both excitatory, you can imagine that would have a deep effect. They're both inhibitory, they would have extra strength. But then if one is excitatory and one is inhibitory, then they can be coming in at different times to kind of say, inhibit this excitatory, or excitatory going past this sort of inhibitory signal. So these various things can happen right next to each other with different neurotransmitters. And then we already talked about the axoaxonic, sort of right here. And the axosecretory, this is where an axon terminal ends on a tiny blood vessel and secretes the transmitter directly into the blood. This is also what we would mostly likely see with hormones here. So you can see all of these different ways, these different types of chemical synapses to basically, in each of these different synapses, they're specialized in location, structure, function, as I said, and they have a target, each of them. And so this is all of these different types of connections are what you can have. All right, so I'm going to go through and give just a little bit of an illustration of this synapse. So, and we had a little bit of the from the example that I gave that was literally the sort of graphical picture, the close-up microscopic picture of the synapse. So here we have the end of the axon with the myelin. Here's the axon terminal, and then right here at this space is the presynaptic terminal button. This is basically the part that would again connect with the dendritic spine over here, or depending upon the type of connection that it had. And so you see these voltage gated calcium channels. So when these open and things and you have shifts in the voltage, then that causes exocytosis, where it basically causes these to open and the neurotransmitters to emerge. You can see this process here, where it will basically be releasing all of these neurotransmitters into the synaptic cleft. And these are the vesicles that hold the neurotransmitters. And of course, here are the neurotransmitters. So, so this is how basically these would be released. They would travel across the synaptic cleft, which is very tiny, but the molecules are very tiny. And then at the end on this postsynaptic dendrite would be these receptors that are chemically gated ions, and that the neurotransmitters will basically connect and sort of attach with some of those, and then activate those ion channels to have an excitatory postsynaptic potential right here, or an inhibitory postsynaptic potential right here. So the cell through all of this can be like, we're going to help make the cell fire more, which is be excitatory, or it could be going through this whole neuronal communication process to be inhibitory and have the cell fire less. All right, here, and we have these postsynaptic receptors up here. So this is what do the receptors look like on this other end of the syllabus. So if we were, excuse me, on the other end of the other end of the syllabus, I'm sorry, on the other end of this dendritic spine over here. So these little pieces, if we go and take a closer look, then we have two of these types of postsynaptic receptors: an ionotropic receptor, a receptor connected directly to an ion channel, and this is fast acting. So these would be up here. This would be the neurotransmitter. It's binding to the receptor, and then basically it lets these ions flow through. And then you can see it here. They're released into the synaptic cleft. They attach to this receptor protein at the end, and that when they attach to this receptor protein, then it causes these the ions to shift and again cause these gradient changes. The final type of postsynaptic receptor is a metatropic receptor, and this is a receptor that affects an ion channel through a second messenger system. And so you can see that basically here, and I'll go through it in a second. Metatropic receptors are slower acting, and they are more common than ionotropic receptors. So in these metatropic, you can see that the neurotransmitter binds, okay? And then what that does is this affects this molecule that then basically has this, G protein that's bound to that. This is basically the secondary sort of messenger piece, and that when these neurotransmitters are released here, this G protein is activated, and it basically then releases, ATP and produces ATP to affect, to basically give energy to this neighboring molecule right here. And then these ions can flow through here. So this is basically a direct connection, through the ion channel. And this is a secondary messenger effects here. And then a message has to be sent over through these here for the channel to open and the ions to flow through. So, and the second messenger, this is the G protein activated and binds to the effector protein. The ATP creates these secondary molecules, the ATP creates these smaller molecules could be other types of enzymes or anything that have a range of effects on these other types of proteins. They can let certain types of proteins through that the cell needs for prime operating function, and they could also be sending messages within, this is a message within the cell from one place to over here to basically be giving rises to cascading changes and stuff like that might also again be the types of things that can cause changes in gene expression and things like that based upon experience. All righty. And this looks like I've got animation set up. So finally, we're going to go over, as I've talked about, then one neuron can basically have this, an excitatory and inhibitory effect on the second neuron that it's connecting with. So these chemical messengers, they're released and they have effects on the postsynaptic neuron. The excitatory postsynaptic potential, this is an EPSP, is a neurotransmitter causing an increase in the likelihood that the postsynaptic neuron will send a signal. Okay, so it's making it more likely to fire. And then inhibitory postsynaptic potential, a neurotransmitter causing a decrease in the likelihood that a postsynaptic neuron will signal. And so a couple of these terms have come up multiple times, but then you can see that you mentioned here, but then as you get to this point, you can kind of understand the fuller meaning of what, you know, an APSP or IPSP would be doing. And so you can see some of these pieces here, how they would, you know, an excitatory pathway, you know, would cause, you know, this to sort of depolarize, you know, but then an inhibitory pathway, you know, would cause this neuron to hyperpolarize more. But this is what you can do to actually record some of these effects on the neuron. And then the other piece that we have is like, well, we have these IPSPs and NPSPs, and they could come from all over the place. So how do we sum them together? So you can see here, it's like, look, these are all these other little, you know, axons and sort of dendrites that are connecting with various parts of this cell. And you can kind of see the differences in sort of the red and the blue, which are going to have sort of excitatory and inhibitory difference, spaces there. And so, but then what happens is there has to basically be the cumulative sum of this information, what reaches this axon hillock right here. And so this cumulative sort of summing occurs through two ways. It occurs through spatial summation, where you have the synapses that are like nearby each other and how they can like fire and have cumulative effects. And then you also have temporal summation, you know, a cumulative effect of many signals, but in a short amount of time. So if you've gotten a number of signals that are coming in from a couple of these different places, they are spatially very different, but they all happen to come in at relatively the same time point, then they will be able to kind of sum through the fact that they're all occurring at similar times. And this is basically the process that will help with that, or help sort of explain some of these pieces. So EPSPs produced at the same time, but on separate parts of the membrane, do not influence each other. They're too spatially far apart, okay? And so you could have the depolarization that would be over here and the depolarization that would be over here, but it's not enough to kind of affect the cell body to reach this axon hillock right here. But if you had EPSPs produced at the same time or close together, then they can add to form a larger EPSP, okay? So here these are nearby. So if they fire together, then they can actually sort of build on each other. And if they fire in short succession, they can kind of build on each other through a little bit of kind of combining the waves. But if they're really far apart in time, then they won't necessarily build on each other. Sort of this would be like a solo of S1 and S2. But if they're nearby and in the same time frame, then they can combine. And then we have this sort of example here of what this would look like, of where you'd have the EPSPs and the IPSPs with regards to temporal spacing and so on and so forth. So if you had a wide temporal spacing, just like we showed, they wouldn't really sum together, and you'd have the same thing for the IPSPs. But if you have close, and again, look, this is threshold, and they're close enough in time, then you can see how they build and get a little bit closer. Similarly, how they would become more hyperpolarized. But then if they're close enough, then they're basically going to go ahead and be able to increase enough to hope, you know, ideally, if the action potential is due, to cause this action potential to meet the threshold and cause the action potential right through here. So this is all these pieces basically happening in the sort of combined example here. So if the EPSPs and the IPSPs on the dendritic cell body charge the membrane of the axon hillock, and action potential is generated and travels down the axon, okay? So here you can see how these would be distributed, okay? So summed IPSPs and EPAs on the dendritic tree and the cell body. So these can sum from down here and have these cascading effects that reach all the way down to the axon hillock. But you can tell that connections up here, they are not going to have as strong as of a connection in illustrating whether or in affecting whether an action potential occurs as right here, like right next to the axon hillock. So these are kind of like, these ions aren't like, they're much more likely to have that effect. And this is based on this spatial sort of and summation with reflect to the, to the axon hillock. Then it depolarizes and reaches down, and you have some of these things continue. So I hope this sort of shows how all of these pieces come together. This is the end of this section, and I will see you on the next section in just a couple of minutes. Take care, you guys.